All right, everybody. Quick heads up too, this is something I'll be sending to the adjuncts who are teaching the lab. Get out of it. Because I didn't have time to talk about the fly linkage assignments. There you go. One. I made a short video kind of basically explaining, what's what. And that video is all the way down here. Right, so it's in the playlist for this section and for the other section too. And I'll be emailing that link round to the adjuncts and to the lab section. So there's absolutely no kind of way that you'll miss it. So if you want to, if you were in my lab section yesterday, hang on, let me unplug my how about now? Testing. How's that sound now, Stephanie? Can you hear me? Ah, good. Cool. Yeah, some reason every so often it tells me my speakers aren't working even though they are. Anyway, I think it was just a link from my cycle pannier and the plug. So yeah, that's up there. Even if you're in a section where there is time to go over that, you can always refer back to this if you need to. Okay, so we are on the last stretch of gene expression. This is pretty much where we ended up on Monday. Actually, we were looking through, pardon me, this video, I do believe. Yeah, that one, right? Which is, I don't know where Dr. Page found it, but it's a very, very good video for explaining the whole process. So really what we have left to do, there's not a whole bunch, but there's, what there is kind of important as always. And I don't know why I'm not seeing these over there. Well. So essentially, as with other processes, initiation, elongation, termination. And termination is all about stop codons, right? So I'm just going to pick one. No. This is kind of annoying. There we go. Third time's a charm. So this is the genetic code, right? So there's 64 possible codons based on the four different possibilities for each of the three positions. And as you can see, most of them, the vast majority, encode amino acids. Three of them, UAA, the ones in red basically, UGA and UAG, do not, right? So there is no tRNA that recognizes the stop codon. And so let's see if I can find my way back. No, this is just going to be a pain in the ass. All right, hang on, I'm just going to save this and reopen it because this is just annoying me. I don't know why it's doing that, but it's just making life harder than it has to be. There you go, yeah, back again, or not. All right, anyway, well, nothing we can do about that, unfortunately. So you're just going to watch me kind of poke around in here until I find the right thing. So, all right, so essentially, ribosome assembles over the start codon, and then essentially the reading frame, right, the words, are specified by the start codon. So every 3 bases after the start codon equals a word, a codon. And then at some point, there we go, found it again. The last codon, the end of the open reading frame, right? Remember, it's bookended on either end by a start codon, an AUG, and a stop codon. That's actually how you find genes in terms of bioinformatics, amongst other things, right? You actually look for those. And that tells you, with some degree of certainty, gene be here, right? Anyway, so at some point, the ribosome will process or kind of move along so that the P site is now over a start codon. And so there's a couple of things going on here, right? One is we have our polypeptide attached to a tRNA that's now in the P site. You can kind of make this invigorated. There we go. Also, because there's no tRNA coming in, there's nothing to add that polypeptide to, right? There's no tRNA with amino acid in the P site, in the A site anymore, right? We just have this captured tRNA in the P site, right? So you have to kind of use your imagination a bit and think about this moving one along. So translation cannot continue, but it's essentially stalled there, right? It can't do anything, right? There's nowhere to put this polypeptide. So now we need another thing, in this case called a release factor. That's what this shape thing is. And it has basically two purposes. One is to cleave that polypeptide from its tRNA. Right? Otherwise, you can't get a functional protein. It will just be tethered there. Right? It will never be able to leave, which obviously is not good. And the second thing is we need to get this ribosome off of the mRNA, because otherwise there's not going to be the space for whatever ribosome is following along behind it to finish translation. Right, so the release factor does those two things. First, it cleaves the polypeptide from the last tRNA, the one in the P site, in this case for asparagine. And second, this is all with the aid of energy in the form of GTP, or hydrolysis of GTP. And then the second is it kind of pushes apart the ribosome and forces it off of the mRNA. So now you have the other small subunit. Oh, there it is. It's hiding behind my chat box. So now you have the mRNA, and this is obviously a stylized version. There will be other ribosomes on it, right? Trying to catch up. But all the different pieces, right? The tRNAs, the various subunits and like, are now free, and they can now, in the case of tRNAs, be recharged. In the case of the ribosome, begin translation again. Right? Essentially, there's a pool of these things which just keeps kind of doing its job, essentially, right? They never stop. And now we have a finished polypeptide. Now, this is not a finished protein. There's an awful lot more that needs to happen before this series of peptides, amino acids in a string, becomes a functional protein. Right. And quite often, the most challenging part is getting this to fold right into the correct conformation. So let's see if I can guess where it's there. Nope. It's there. There we go. Yeah, it wasn't too bad. So this complex three-dimensional shape does not happen just by chance. It's often aided by the essentially the presence of other proteins called chaperones, right, that help that protein form the correct conformation. I have to use some of my memory powers here to remember which slide I was Okay, so that's pretty much it, right? So that's the basics of translation. Now, as I was saying earlier, there's not just one ribosome translating an mRNA at any time. And in both eukaryotes and prokaryotes, as soon as a sufficient bit of the mRNA has been translated, right, so that AUG, the start codon, is free and hanging in the breeze, then a second translation initiation complex can begin, right? And so you can have multiple, obviously depending on how big the mRNA is, and also how efficient initiation of translation is, right? It doesn't happen the same for all transcripts. Some are translated much more readily than others. But despite all of that, you will have multiple ribosomes translating the same mRNA, right? So as one is finishing, right, so we have our polypeptide released here, and this ribosome dissociating, the aid of the release factor, now we have another ribosome assembling to begin translation. And all the way along, you'll see essentially polypeptides in different states of synthesis. This is called a polysome. Now, so far, other than talking about initiation of translation, right, the scanning in eukaryotes versus the assembly on the start codon for prokaryotes, most of this is more or less the same. Right. Now, one really big difference, and this is actually key to how prokaryotes control gene expression for many operons. Ooh, actually, I haven't introduced operons yet. So, two big things. This is where not being able to see kind of what I'm doing is a bit of a challenge. So the first one is that genes involved in the same process, like say a metabolic pathway, are grouped together into one regulatory, I can spell that right, transcriptional unit. That is called an operon. And so it's quite a nifty idea if you think about it, you know, because if you have a particular metabolite that you want to use, then you need all of the things that are required to use that metabolite, right? So all of them have to be turned on or produced. If you don't have that metabolite, then you don't need any of them, right? So it's easier just to kind of turn things on and off in blocks, right? And don't forget as well, in bacteria, gene expression is on by default. It has to be turned off. And those genes, when transcribed, form one large mRNA. If I can spell that right, with multiple coding sequences. And that is called a polycistronic mRNA. Second big deal about prokaryotes is the transcription and translation are coupled. So as soon as start codon, emerges from the RNA, from the RNA poly. A ribosome assembles and begins translation. That is also another key way in which prokaryotes control gene expression. And we'll be looking at that in some depth with the TRP operon, which will be in the next section. So this is what a polycistronic mRNA looks like, right? So you have start or the beginning and the end, and in it you have multiple different coding sequences or open reading frames, each bookended by their own start and stop codon. Right, so this is one continuous mRNA, but it produces 3 distinct polypeptides. And so essentially, it's like producing the whole packet, right? And from that, you can make all of the proteins that you need. In eukaryotes, almost always one gene is one mRNA, right? So they're monocistronic. Gene expression in eukaryotes is actually controlled or coordinated in a different way. Rather than by grouping them together, it's done by production of specific proteins that help transcription of similar genes. And so another reason why transcription and translation can be coupled in prokaryotes is because they're physically occurring in the same space and therefore can occur at the same time. So eukaryotes, you can't do this because the translation machinery is physically separated from the transcription machinery by the nuclear membrane. So even if one wanted to, you could not, right? Which has its own benefits, right? It's not like we need to be jealous of prokaryotes, but there are pluses and minuses both sides. And so this is kind of, actually, this is a, what would that be? That would be a transmission electron micrograph of some bacterial DNA. And these big blobs, because they are actually physically large, are the ribosomes. And so essentially you have one piece of DNA, multiple mRNAs, and each of those mRNAs forms a polysome, right? And so you can see how this can lead to a very quick expansion of protein production, right? Because you don't have to wait for this mRNA to be finished to be translated. You can already be translating it before you even finish transcribing it, right? And so kind of to put this into the bigger context, you know, bacteria are not able to, well, not really, manipulate their environment, right? So they are at the mercy of any changes in their environment. So in order to survive, they need to be able to essentially respond to those changes very quickly, right? They can't store a lot of stuff, right? They're small and, you know, you don't want to waste energy because energy equals survival and reproduction, essentially. And so you want to be able to have things off you don't need. And then when the environment changes, have a lot of that stuff, right? Whatever that might be. And so bacteria are able to ramp up gene expression, production of proteins very quickly to be able to take advantage of changes in their environment. It's much slower in eukaryotes because there's a lot more that needs to be done. Before we start on that, does anybody have questions about transcription or translation, or RNA processing, or anything that we have covered so far? Because we have a bit of time, and also I'm quite aware of the exam is on Friday. So if you do have questions, now would be a great time to ask them.
Will you still be available before the exam or during that time, like 9 to 9:50?
Yes, I will be. Yeah, I'll log on to the Zoom, and I'll be checking my email and reading the news and stuff like that. But yeah, if you have any questions about the material, right, obviously I'm not going to tell you the answer while you're doing the exam, or about the exam itself, then that would be a great time to do so. Yeah, absolutely.
Oh, sir, the chapters for the this exam, I just wanted to clarify. It's from chapter. Hold on. Chapter. Hold on. Let's see. From chapter 12 to 17 or 18.
12 to 16.
Okay.
So genome structure. I don't know why I'm not hearing you through my headphones, but hopefully you can hear me okay. Yeah, so it's from 12, which is genome structure dynamics, through transposons. Ignore 14 there, it's a bit of a relic. And then 15 and 16. So it's 12, 13, 15, and 16 are what you will be quizzed on. And obviously what we covered in these sessions and what's in the chat records that are posted to Blackboard too. Which really
I had a question about the chat. So the 22nd of October, that one isn't posted. I was just reviewing them the other day, but okay. Yeah, so I was just gonna let you know it's not on there.
Let me just have a look. Hopefully I actually have him. Sometimes funky stuff. Oh yeah, 27 October. Let me get out of it. Come on, push. It's just there. All right, let me just get that uploading while I'm talking, because I've got memory of a fish and also got an exam to write as well. So I don't want to forget that. Yeah, my apologies. I'm usually pretty good about get out of it, uploading these things, but sometimes I miss it for some reason. Hopefully let me just double check YouTube link as well, make sure I've uploaded the yeah, weird. Uploaded the YouTube video, but I just didn't upload the chat. Anyway, thanks for that. Appreciate the heads up. Okay, anyone have any other questions? All right, so essentially what I want to do now is talk a little bit about kind of the genetic code, a little bit about what's called degeneracy, right? And so not that I'm degenerate, but we'll talk about the degeneracy in the genetic code. A little bit about wobble, and also a little bit about mutation, right? So kind of this is tying back to what we talked about a ways ago about different types of mutation. Right, so essentially, come on, don't talk about that just yet. Each codon is recognized, other than the stop codons, by a tRNA. Now, some codons are, well, get it the right way around. Some amino acids, there's only two, methionine and tryptophan, are recognized. They basically are encoded by only one codon, right? So these are unique. I'm pretty sure there's no tryptophan anywhere else. Let me just double check. Yep. So there's only one tRNA and one codon for methionine, right? That's the start codon. You don't actually tend to find methionines kind of scattered around in the midst of a polypeptide. They seem to be largely reserved for the start, right? So the AUG codon is always the start codon, right? If this is mutated, no protein, right? Because the ribosome has no start point, essentially, right? So it never begins translation. However, a lot of the other amino acids are encoded by multiple codons, right? So leucine is actually the kind of record holder. There's another one which has 6 as well, I think. I'd have to have a good solid look through it. Oh yeah, serine, there you go. Anyway, so leucine and serine can be encoded by 6 different codons, right? This is called degeneracy, right? And what it means is that some of the letters in that codon are not important. And so if you look at proline, for example, this one here, so you read this table from the left first and then the top second and then each individual, right? So if you wanted to look for AUG, it's A, U, and then G. Hope that makes sense. Anyway, in proline, all that matters are the C and the C. Haha, Pirate's favorite letter. Oh. Awesome. I just remembered a cool dad joke that my daughter told me the other day. Why can't melons marry? It's got melons. Anybody? Why can't melons marry? You ready? Because they cantaloupe. 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 Ah. Well, I thought it was funny. I know, isn't that terrible? I'll just type out posterity. Anyway. Yeah. Anyway, back to the program. And so that means that third letter, third base in the codon can change. So if you have a coding region or an open reading frame which has a CCC, right, which encodes proline, if that is changed to a CCU, nothing happens, right? That is called a silent mutation, right? Because there is a mutation, right? There's a sequence difference between that and, you know, the same gene in a same species, but there's no functional difference in terms of protein. Now, if you change CCC, right, to UCC, now you have serine, and that is different. Actually, let's see if I can dig out where, which one was I on for a start? 39, okay. Ooh, close. There we go. So proline is actually, it's a bit of a funky amino acid. It kind of forms a ring shape. It's hydrophobic. It's a reasonable size, I guess. And serine is hydrophilic, right? And so changing from this amino acid to this amino acid, see, depending where in the protein it is and how important that residue is, will almost certainly disrupt protein structure and or function, right? So there is some degree of buffer, right, in the genetic code, right? Basically, you've got a 1 in 3 chance, depending on the amino acid, of that mutation not having any effect. And that's basically caused by there not being as many tRNAs as there are codons, right? So there is one tRNA that recognizes CC. And then the last, and this is called wobble, right? The third base of the codon is less important, right? And that's caused by basically there being a less important contribution, essentially, of the third base pairing, that pair in the codon, towards that tRNA coming in properly. That's called wobble. So we have a degree of degeneracy, right? But there are some mutations which are catastrophic in terms of translation and protein production, gene function. Obviously, changing the start codon, and this is what we will see in our yellow molecular biology project, right? So the start codon has been changed in the yellow allele, mutant allele. So that's obviously not good. No translation, no polypeptides. But the reading frame is dictated by the methionine. Come on. It's up here somewhere. Here we go. So the ribosome has no way other than the start codon of knowing what the reading frame is. And by reading frame, I mean the series of codons which encode amino acids. So the ribosome, all it knows is where the start codon is, right? And in case of eukaryotes, it knows it because of the base pairing of the FMET tRNA. In prokaryotes, it's based on the Shine-Delgado sequence, essentially bringing the small subunit in, right? And so every 3 letters after the start codon equals 1 amino acid. That's the reading frame. Now, so essentially, let's kind of blow this up a little bit. So on slide 28, reading frame, this is where working on the board would make things easier. Let's put this over here. Equals AUG, obviously, right? Because that's the start codon. Then UUU, then GAA, right? So I'm reading these, like the dashes are just to separate the words, right? There are no dashes in here. And so I'm just reading it in groups of 3. GAA, GUG, AGU, and so on. Does that make sense, everybody? Give me a shout if it doesn't. I'm working off this sequence just here. Now, let's say, I'm just going to work this out. Now, Now, if we delete that third U, right? So this one here, right? So if there's a mutation which removes a base, that's called a deletion mutation. You can also have insertion mutations as well, where you add bases. Now our reading frame is, again, AUG, right? Because that's the star codon. We haven't changed that. Now we have two U's, because we've removed, see that one that has a little bubble underneath it? That has been removed by the mutation or some kind of chemical mutagen, which has been, resulting in being excised by base repair, stuff like that. Now our reading frame is UUG, AAG, right? Because again, let's see if I can actually. Come on. Oh, here we go. That's good. You want that. There you go. That's kind of clunky, but it'll do. Right, now it's UUG, AAG. Right, so already, let's see what UUG encodes. Where's my, is that 39? Yeah, hey. All right, so let's go back out. UUG. So we've gone from phenylalanine. So it was methionine, then UUU is phenylalanine. What was the next one? GAA, GAA, glutamine. No, glutamate, sorry. Always get those confused. Right, that was the original reading frame. Now it is next. UUG is leucine. What's AAG? AAG, lysine. So now you can see, just by removing one base, we've changed the reading frame after the methionine. And so if we change the reading frame, we change the series of three-letter words, and we change the sequence of amino acids in that polypeptide. Right. This is called a frame shift mutation. One, A equals a nonsense protein. Actually also called a nonsense mutation for that same reason. Okay, so let's go back, if I can remember where it was. Oh. What's up? Did someone have a revelation or a conniption? Okay, so what was our reading frame again? Okay, so it's new reading frame is AUG, UUG, AAG. Right. What's the next codon? Right, so it's UUG, AAG. What's the next one? Just here.
Wait, so wait, where?
Let's go back to,
Wait, where?
it's going to be a UGA, right? And what does UGA encode? Anybody find that one for me on that genetic code chart? What amino acid does UGA encode? Yeah, thank you, everybody. Sounds like someone's got a car alarm going off in the background. It doesn't encode an amino acid. It's a stop, right? So the second thing that a frame shift often mutation results in is a new stop codon. Because that stop codon wasn't originally in the frame, right? So it doesn't stop the protein, right? The ribosome just reads straight through it because it doesn't kind of exist, right? We're not reading the three-letter words in that particular way, right? We're in a different reading frame. So if we've shifted the frame now, just by random chance, as actually we see there, so this semester was the first time I've seen that, which is kind of curious, right? It was not intentional. Now we get a stop codon. So frame shift mutations, if I can type it out, right? Result in, pardon me, a nonsense protein, at least, and usually a new stop codon and a truncated protein, both of which equal no function, right? So that frame shift mutations, as opposed to mutations in the third base of each codon, frame shift mutations are almost always loss of function mutations. Right? Base changes in the third base may cause problems. So for example, if you had a UGU codon and that U changed to an A, well, now you have a new stop codon, right, which is bad. But for others, leucine, for example, you could change the third letter to whatever you want, and it's not going to have any effect on the protein or the amino acid sequence, and therefore the protein. So the consequence of mutations very much depends on not just where they occur, but what they are as well, even in the open reading frame. Cool, and that's pretty much all I've got from today. And that's finished us up quite tidily. Anyone have questions about anything, pretty much? All right, well, thanks very much, everybody. Have a lovely day. I'll see some of you in an hour and a bit. And I'm also, I presume Mr. Penny has already told you this, but for those of you that are in his section tomorrow, I'll be hosting the lab tomorrow as well. So you'll see me there and not Mr. Penny because he is quarantined. Actually, funny fact, some of you probably already know this, the word quarantine, which is kind of topical now, right, actually comes from, I think it's Venice, many moons ago, or Italy in general, where ships that arrived, like sailing ships that arrived with, diseases on board, had to stay offshore for 40 days. And that was, I can't remember the quarenta is Spanish, but the equivalent in Italian basically means 40 days, right? So that's where the word quarantine actually comes from. This is a period of time in which those ships were made to stay offshore so they didn't bring the plague or whatever nastiness with them. There you go. Fun fact for the day. Cool. Have a lovely day, everybody. I shall see you all soon. Work hard and good luck in the exam.